The migration of life is sort of the topic. The question, of course, is what is life? And life is a self-sustaining chemical system capable of Darwinian evolution. We already have one example of it, and those are the UR-backs that you have already heard Gary described you, all related by common ancestry. You are, of course, the U, the R are the archaebacteria, and the bacteria are the eubacteria or bacteria. And what's quite clear is that you're going to get a second example of a Darwinian system, not from searching for life, but rather from generation in the laboratory. And the reason why you don't have that quite already is because of two things. First, it's actually quite difficult to make. Pretty much everybody relies on molecular biology tools that you can buy from Sigma, Aldrich, New England Biolabs, Promega to do manipulations of complicated molecular systems. The problem is that enzymes that have evolved for 4 billion years to do natural DNA don't generally do unnatural DNA or unnatural systems very well. And so this creates the need to do from bottom-up redevelopment of all the analytical chemistry, all the synthetic chemistry, all the molecular biology, and all the cell biology that you take for granted as a part of the of developing an artificial life form. And that takes time, and it takes hard work, and it takes the need for funds. But let's just see if I can get this to work now. But of course, there's a second reason why we don't have this already, and that's because self-sustaining is seen as a threat. And this is actually an article on the right-hand side from the New York Times, Andrew Pollack, when our buddy Floyd Romersberg actually got 6 letters of different DNA to work inside of an E. coli cell. And Andrew Pollack wrote that it raises, that this adding letters to DNA's alphabet raises hope and fear. And we are urging a convening of a commission of scientists and the agencies that fund them, right, to discuss whether or not some new guidelines for work should be using, some new lines of work using modified genetic codes should be suspended because they're afraid that this will take over the planet. Now, in fact, the search, we pursue the search for alien life because we actually want to learn more about what life is in general. A second example will be very informative of this. And of course, it also helps us pursue or search for or recognize when we found it off-Earth alien life. And this is actually how chemists think about universals. It's not quite what you heard this morning from Sarah, where you're talking about sort of hypothetical information things. Chemistry is actually hard to get to work. The molecules precipitate, the molecules hydrolyze, the molecules decompose. And so it's very much a constraint that you have to deal with when you're trying to create artificial genetic systems. So what we have here right now is on the picture is 2 sets of structures. These are the so-called 8-letter Darwinian systems. It's Hachimoji because the lead author on this paper is Shuichi Hoshika, who's Japanese. And for those of you who know Japanese, you know that Hachi means 8 and Moji means letter, as in emoji. And so these are 8-letter DNA. They fit the laws of life as we know them from a chemistry perspective, as they have 2 features. One of this, the so-called Benner criterion for genetic biopolymer, which is the polycharged backbone, is necessary so as to make evolution possible. You can mutate the sequence of letters in this DNA without changing its physical properties. There's also the so-called Schrodinger requirement, which you heard about this morning as well, which is that the building blocks must have not a physical chemistry constancy, but rather a structural constancy. That is, all the building blocks have to fit into an aperiodic crystal structure. That's what allows them to evolve. And so the 8 building blocks form 4 base pairs now, and so unlike the 4 building blocks that form the 2 base pairs in UR-back DNA and RNA. And you got to keep in mind that I won't have a chance to talk about it, but all the hard work is in the synthetic and analytical chemistry, the developing of the enzymology for these and all the rest of it. And as we like to say, now that I can use Gary's use of language as one goddamn problem after another in that side of it. But never mind, the bottom line is the 8-letter conformity to the Schrodinger requirement is actually quite remarkable. You're looking at crystal structures that were developed by Millie Georgiadis, where you can see in B 4 different sequences superimposed upon them. C, D, and E are listed on the board, which are 8-letter DNA superimposed upon the 4th, which is just standard DNA. They're basically superimposable. And what that means is you can mutate from one to the other without changing either their physical properties or their structural properties. And that means that you can evolve the system. And of course, this system could then be what Gary asked me to do, which is forget about it, looking for aliens, just make them in the laboratory, and that could be the topic of the talk. But and there's actually some reason to do that, because although Hachimoji DNA is not self-sustaining, we don't build that in. It meets other 4 requirements for life. For example, it will store information predictably, and that's what the rules are in the lower left-hand corner of this plot. It's just a statement where we've gotten melting temperatures for hundreds of DNA duplexes so that we know how much a TA pair contributes to a duplex stability, or a PZ pair, or an SB pair, or a GC pair, and it's quite well predictable. So that's the first requirement. Second, the information in the DNA can be transferred to another biopolymer, in this case, Hachimoji RNA. The second system has a selectable phenotypes. Here it was fluorescent, and what you're looking on the right-hand side of that slide is actually the Hachimoji RNA that binds, folds, and binds the fluorophore like spinach. The aptamer does with just 4 letters, and it glows green. It also, by the way, for Harry, works in a ribosome to encode 21, 22, 23 different amino acids. Because of the informational structural similarity of the informational units, they're freely interchangeable. They can evolve. Now, however, this is actually a Science News, thescientist.com editorial from a couple weeks ago saying this newly expanded genetic alphabet that includes 4 synthetic nucleotides highlights the need for strict boundaries on its use. Should there be a genetic firewall, do we want to worry about our descendants who see who wins and loses, where your DNA is going to be competing with our synthetic DNA? Now, for those in the laboratory who actually struggle to keep this thing alive, we know that that's a problem. But it raises an interesting question, and that is whether alien life forms coming from someplace else that invade an pre-existing, pre-adapted biosphere, will they survive like the Burmese python does in the Everglades, right, as an invasive species, or will they die, which is what would happen if I dropped you into the Everglades, even though you're an intelligent person? And that's a very interesting question. It's a very general question. Now, there's a problem, and that is that these lab-generated aliens are uninteresting, okay? You are nowhere near as excited about laboratory alien DNA as you would if you were to encounter this guy, Spock. That's a cultural immutable. I could stand here all day and try to persuade you should not be more interested in aliens on another planet than you are in our stuff. It will not work. In fact, this is a poster that's hanging in the wall of my office, given to me by my sister. I am much more excited by Spock, partly because he can be the product of a male Vulcan father and a female Earth mother. That is a remarkable statement about mutability and universality. At that point, my wife will say to me, "Stephen, dear, what part of the word fiction don't you understand?" Right? But it does help us look for alien life, especially should it not share our you are back molecular biology, and that's what you heard from Stephen Hawking this morning. And it might help us understand the migration of life, because the migration of life involves 2 rates. Right? One is the rate at which life emerges, and the second is the rate at life migrates after it emerges, as David Catling was pointing out a little while ago. So if the first rate is slow, the fast second rate might compensate and spread life throughout the galaxy. But if the first rate is fast, and the migrating life, when it does move, will encounter a pre-adapted biosphere, for which only intelligence can compensate for the fact that the life in the territory that you are invading already has a head start. And that, of course, includes our alien life. If our alien life form gets out into the San Diego metropolitan area, will it get to the zoo and eat the penguins, right? Or will it will fail because that life has already been around in that environment for a long time and therefore has no reason to evolve. So we actually have some idea when and how life arose on Earth under this so-called RNA-first model that you heard about. So, for example, we know that this molecule, hydroxymethylsulfonate, which I actually brought a sample of here, actually is present on the early Earth, okay? You can ask, how can we possibly know that? You may keep that for safekeeping, don't eat it. If you open it up, alien life might come out and take over the planet. So how can we possibly know that? Well, because we know 2 things about the early Earth for sure. One is it had a lot of volcanism, and second, that's the volcano that you're seeing. And then second, below you can see it is generating a lot of formaldehyde. And this hydroxymethylsulfonate is simply the addition product of formaldehyde and sulfur dioxide. Now you can ask the question, okay, well, how do you know that the volcano is generating sulfur dioxide, but not, say, a more reduced form of sulfur like hydrogen sulfide or a more oxidized form? And the answer goes back to some work that Dustin Trail did with some 4.3 billion-year-old zircons, where he looked at the ratio of cerium-3 and cerium-4 entrapped in those zircons and concluded that the mantle from which those zircons crystallized 4.3 billion years ago must have had a redox state suitable for the evolution of sulfur dioxide. So this is what we are calling bespoke or privileged chemistry. You cannot avoid it. On an early Earth, there was no question that you were generating formaldehyde. That's what the plot is on the lower left of this plot. And the formaldehyde generation is largely independent of the details of the atmosphere that's being generated from. It's being generated by lightning or ultraviolet irradiation of a carbon dioxide-rich atmosphere with various methane carbon dioxide ratios. But the fact that it's being formed and sulfur dioxide is being formed, so you must have that compound that you have sitting over there in that corner. Now that's good, because unlike formaldehyde itself, which reacts with itself to form useless products, this hydroxymethyl sulfate is unreactive. It's a reservoir for carbohydrate that bleeds formaldehyde into a prebiotic soup where it solves many, many problems in the synthesis of RNA. And by the way, this is different with the next higher carbohydrate. This is a 2-carbon carbohydrate, glycolaldehyde. This is the same plot expanded now with glycolaldehyde put onto the plot. You have to remember that there's a millionfold change in scale. That is 1/1,000,000th of glycolaldehyde is actually being made relative to formaldehyde. But when the methane formaldehyde ratio drops to 0, the formation of glycolaldehyde drops to 0 as well. So these are universal facts. They constrain models for the formation of ribose in any RNA model to gain access to Darwinism. Glycolaldehyde, because it is made in 1 part per million, that amount of formaldehyde cannot participate stoichiometrically in any pathway for the formation of ribose. It may participate in this process catalytically. Indeed, the catalytic participation is bespoke. One cannot avoid it on any Earth-like planets, even if one wanted to. In fact, it's probably going on right now on Mars because all of the components are there. So you can probably see this reaction taking place on Mars. So you can now choose any molecule pathway to a model that you might like. So Matt Palner, about 10 years ago, offered a great model which you can choose. Unfortunately, it requires glycolaldehyde in the upper left-hand box shaded in magenta stoichiometrically. Okay, maybe the Hadean had it, but you would have to have some explanation for that proposition. But if you don't like Matt's model, you can use ours, right? Here we have the magenta glycolaldehyde in the lower right-hand corner of that slide, and that actually is catalytically formed. And that's come from 2 arrows that allow formaldehyde to be fixed in a cycle that leads to the formation of ribose. And so you cannot avoid that particular cycle. This particular cycle is probably taking place today, for example, on Mars. On Earth, of course, any of the things that you make in this cycle would be eaten by the life that's already here. So we can put together this so-called bespoke chemistry that makes carbohydrates, which are CH2O to a power N, as are inevitable. And what you're actually seeing here is the interaction between formaldehyde and sulfur dioxide in the middle of the left, and then you're seeing the interaction of glycolaldehyde, which also forms an adduct with sulfur dioxide to become a stable reservoir. And you're able to get these to process to sieve 2 and 3 and eventually 5 carbon species, which we've drawn on the side. Now, I've actually put up here also the minerals. I won't mention much of them, but I did bring one of them along. This is one of these borate minerals that's very valuable, and it's also quite useful because it's also certainly present on Earth where the mantle is sufficiently oxidized that it is producing sulfur dioxide. And the borate minerals are indicated on this slide at various points of catalyzing certain steps or stabilizing certain products. But this makes it, this chemistry is inevitable. And of course, if this chemistry in its inevitability means life is more likely to emerge, this means that when it is not necessary for it to migrate, and maybe if it does migrate, it will already enter another habitat which already has a pre-adapted, pre-existing life form occupying the space. Now, keep in mind that while the carbohydrates then are relatively easy, we are in trouble with respect to reduced nitrogen, okay? And we are, so no Titan, for example, and of course we're also in trouble because large amounts of carbohydrates are raining to the surface. Titan is a model okay? That is, the organics on Titan are raining to the surface. But the problem is that if the chemistry of the atmosphere has the oxidation of sulfur dioxide, can you also get things like hydrogen cyanide, which David Catling was mentioning? And of course the question is, who cares about hydrogen cyanide? Well, the answer is everybody, because hydrogen cyanide is needed for the bases, also cyanoacetylene, HCCCN, also cyanamide and other reduced organic compounds. And of course with no bases, no RNA, even with bespoke carbohydrates. So the problem with hydrogen cyanide is that just everybody thinks it has it, but the argument always for having it was coming from Leslie Orgel, who noticed hydrogen cyanide in microwave spectroscopy in star-forming clouds. Now, of course, the fact that it's in star-forming cloud doesn't mean it's on Earth, and so people are looking for other ways of getting it. And one of the problems is if the atmosphere is in redox equilibrium with sulfur dioxide, which is needed to stabilize the carbohydrates that I just mentioned and delivered over here, it produces actually very little hydrogen cyanide, cyanoacetylene, and very little cyanamide. And so we looked for other ways of getting this. So, by the way, we need lots of these. This is actually Matt Pauner's molecule, where I've added to here all of and now the blue box is the cases where you have to have these reduced carbon atoms, molecules, and reduced nitrogen molecules. If you cannot have them because the atmosphere is too oxidizing, then you cannot have RNA. And so and all the carbohydrate reservoirs that we make are worthless if there is none of these reduced nitrogen-containing molecules to make the bases. And so what you're seeing here now is a problem which we're going to aggregate now and put on top of each other all the things I've spoken about, right? So obviously, as you go forward in time, you have an increasing amount of carbohydrates. Of course, the carbohydrates have to land, on dry land, so as they don't get diluted into global ocean. We don't know much about the history of dry land in the Hadean, but we assume it increases as time goes forward. And of course, therefore, the amount of carbohydrates accumulating on dry land will be sort of like the product of these, the amount you're making and the amount of dry land under which they can fall. But if the production of bases is zero, you got troubles. So one of the ways we can address this is by looking at another conundrum. This one is from geology. And that is that when the Earth was formed and still molten, the iron sank to the core, taking with it all the iron-loving elements like palladium or iridium or platinum or gold, and yet you can still mine all of these today. These siderophiles were delivered to the crust later after the core sank, and this is the so-called late veneer, by impactors that are large enough to have their own iron cores and large enough to reset the radiochemical clocks in the crust, but not large enough to remelt the Earth. And the data to support this comes from the composition of the late veneer on Earth as well as the distribution of the veneer on Earth versus Mars. So what happens when a body roughly the size of the moon with its own iron core hits the Earth? Well, it resets the clocks in the crust. This happened maybe 4.47 billion years ago, and you heard a little bit about that earlier today. It destroys any life in the crust. Okay, any life that was there must start over. It destroys the organic reservoirs that may have accumulated. You have to start over then as well. But then it dumps tons of molten iron into the atmosphere, and what the molten iron does is it converts the carbon dioxide to methane, then dinitrogen to ammonia, the water to dihydrogen. We may have had 90 bars of hydrogen, I think is a better estimate here. And this is the atmosphere that now can generate hydrogen cyanide, cyanamide, and cyanoacetylene, maybe up as much as 9% upon UV irradiation and lightning. So the same impact that delivered the iron, the gold, platinum, and iridium that we mine today from the late veneer put a reduced atmosphere over an oxidized mantle. It makes the bases able now to be made in the atmosphere to rain out onto the dry land and react with the carbohydrates that are present from the sulfur dioxide reservoir, which I've just handed out. Well, that's kind of great, but of course that's the model which says, okay, we have an impact that produces the late veneer, but also makes the atmosphere reductive, so we can now make large amounts of bases. But that actually will decay over time because the hydrogen will escape from the atmosphere. And so the productivity of the atmosphere, the window of opportunity to make the bases for RNA goes away, maybe with a half-life of about 40 million years. And so the total amount of carbohydrates on dry land is still going up, the amount of dry land is still going up, and maybe the amount of cyanoacetylene and hydrogen cyanide form is going up. But over time, the ability of the atmosphere to make these reducing base precursors goes away. And so what you now can do is combine all those together and get a statement that says the most probable time for RNA formation on Earth was about 4.36 billion years ago, plus or minus about 0.05 billion years ago. And that's when Darwinism is supposed to appear. The interesting part about that is that if you actually go back and look at some of these old zircons, Beth Ann Bell, as we've already heard a few minutes ago, isolated a zircon with a 4.1 billion year old date, which has a carbon-12, carbon-13 ratio, may be indicative of life. And that gives you 250 million years from the time when the formation of RNA was most probable to the time where Beth Ann Bell has the very first evidence that there was an extensive biosphere on Earth. 250 million years to get that biosphere out of origins. And that you might decide is a reasonable time or an unreasonable time. That's up to you. I'm running out of time, so let me not go through phosphate, but let me just say that we have actually perfectly good story because the phosphate also exists on the Earth from an oxidized mantle, so it's in the form of phosphate, not something with lower redox potential. And it turns out that with various minerals associated with it, including the ones that I handed out over there, not only can you make the phosphates of the nucleosides, but you can also make the nucleosides from a species that Ram Krishnamurthy proposed, and actually phosphorylate them not just on the not just to make the glycosidic bond, which is the first important thing from phosphorylation, but actually the diphosphates come out of these borate minerals that are not only stabilizing the nucleoside sugars, but also causing the nucleoside sugars to become diphosphates. This is actually kind of interesting because this is work that has also been reproduced now by Sidney Becker in Tom Carell's laboratory. So there's the schematic of bespoke chemistry leading to RNA. Okay, there are minerals now, opals, bicarbonate minerals that bind and stabilize RNA, all bespoke in a Hadean crust, an optimum formation about 4.36 billion years ago. Now, let me conclude by saying that people express surprise at the precision of that date, but you have to understand the precision is driven by a logic. That is, the most resilient chromophores show no resetting after about 4.45 billion. To be large enough to deliver the veneer, the impactor must reset those clocks. But if the veneer is delivered by non-resetting smaller impacts, you cannot account for the paucity of the veneer on the moon. You have to rely on one or two large impactors hitting the Earth to get the statistics of small numbers to account for the lack of the veneer on the moon. And therefore, the impactor that delivered, okay, and if the veneer delivered, the veneer was delivered earlier, then you have to ask the question, what reset the clocks later without delivering a veneer? Well, thus the impactor that delivered the veneer is about 4.45 billion years ago. Now, that impactor must have been big enough to have its own iron core. The core must have reduced the atmosphere, absent a direct straight-on impact. The physics of atmosphere, the reduced atmosphere, carbohydrate and base formation is bespoke. It's got to happen. The physics of the atmosphere hydrogen escape is relatively well constrained. The restoration time is maybe 20, 40, 60 million years, but it's not 100 million years. And so that is why the window of opportunity closes before 4.3 billion years. You can't make RNA afterwards and be consistent with the geological record. Of course, unfortunately, the big uncertainty is the availability of dry land to collect the material. I just put here all of the various models that are associated with the formation of dry land, and they go from, this is the time scale is in the reverse direction here. The oldest time is to your right, but there are some models that say that we had about the modern level of dry land quickly, some very slowly. But this uncertainty is what's pushing the most likely time of RNA emergence to later in the opportunity window opened by the veneer creating impact. So the notion of life originating on Mars, which you heard about a minute ago, is a way of solving the dry land problem that we can't. So you have to decide first and foremost in this decision tree whether you believe in the RNA first model. If the answer is no, you have a different set of problems. If the answer is yes, you have to make RNA pre-biologically. Okay, you can pursue Benner's stupid chemistry. If you decide not to, my response is to get your own goddamn chemistry. If the answer is yes, you need an oxidized minerals, maybe a desert, dry land. Can we have a dry land on early Earth? Well, here's the point. Some people say, no, the early Earth was a water world. There was no dry land. And that's why you have to go to Mars, because everybody agrees there was always more dry land on Mars. But if you don't accept that, if you think there's some dry land, then you don't need to go to Mars. You can have life on here on Earth. So Mars is unnecessary. if the Hadean had adequate dry land. So there's the overview schematic. This actually was put together by a group of us in Atlanta. This is one of the meetings that Lindy was mentioning where everybody gets together. You don't really know what you're doing when you walk in, but when you walk out 4 hours later, you have a view of an impact that's occurring at a particular time, constrained in its size by the veneer, constraining its dates by geological clocks, constraining chemistry to allow a window of opportunity to make RNA bases to occur. That window disappears over time, but during that time, stuff rains down onto whatever dry land is available. More dry land occurs as it goes along. You have these rocks at the bottom, which are being described from the oxidized mantle, which are stabilizing the RNA. And as sometimes the brown turns to green, as Darwinism forms with a maximum probability at 4.36 billion years ago. And if you believe Beth Ann's interpretation, I think her result is correct, that the interpretation is open. You have now 250 million years to get a biosphere on Earth. With that, let me stop. Thank you for your attention. I must be over time. Uh-oh.
Regarding your dry land problem, Carl Woese made a suggestion a couple of decades ago that may resolve this problem and another problem, which is the formation of cells. He suggested that at the time of the origins, there was the atmosphere was a huge aerosol, and so you could have droplets containing the carbohydrates and at the same time concentrating them as they evaporated and so on.
Yeah, I know nothing about
forming cells
but
without membranes, but then as soon as you made lipids, they would of course coat the outside of these things.
Louis Lerman tells me all about aerosols. David Deemer tells me all about lipids. So let me defer that question to them. But yes, I'd be very interesting to hear more.